So next up we have Ashley Thompson with the complex genetics of TBDs, need for integrated personal and family history, considerations around screenings, and the implications of family planning. Genetic counselors are the instrumental part of the TBD care team. Today we are joined by Miss Ashley Thompson. Ashley recently graduated with her genetic counseling degree. She is also equipped with strong research experience and expertise in clinical genetics from her work at the National Cancer Institute on the genetics of inherited bone marrow failure syndromes and its psychosocial team. We are excited and grateful to have you, and I will give it up to Ashley. Thank you.
All right. Wonderful. Thank you, Kelly. Hello, everyone. I think it's really neat how things kind of come full circle. Katie and I were just talking this morning. We met for the first time 5 years ago, actually, at a basic science telomere conference. And so now, 5 years later, I'm here, genetic counselor, and it's really just neat how things come full circle. So I'm really glad to be here with you all, and it's nice to see all the gear. I actually wore this shirt to a doctor's appointment of my own one time, and they asked me at the end, so a telomere, what do you do, and what are those? So I had the opportunity to share with the physician. So anyways, nice to see all of you all, and I'm excited to kind of go over this complex topic, but I want this to be very interactive. So if you have questions, please stop and ask, because sometimes, or especially with the genetics of telomere biology disorders, there's a lot to take in. And so if you have questions, if something doesn't make sense, please stop me and ask. And I'll also be around this weekend, and we can chat if you have further questions. All right, and so before I get started, though, I do want to know my audience just a little bit. And so I want to know how many of y'all have interacted with a genetic counselor, or potentially seen a genetic counselor, just so I can get a better sense of my audience. I was hoping that would be a lot of hands. Great, perfect. So I may go over a lot of information that you already know. Maybe I'll clarify some that you had questions on. If I'm telling you things you already know, just bear with me. All right, so we'll kind of start from the beginning, though. So what are genes? So we're all made of cells, millions, trillions of cells. And within all of these cells are the nucleus that contain all of our vital genetic information. And those will be packaged into our chromosomes. And so here are chromosomes, and that's what you hear a lot about. 23andMe, the 23 comes from, we have 23 pairs of chromosomes. So we get 23 from our mom and 23 from our dad for a total of 46. And so we have our chromosomes, and our chromosomes are really just tightly wound complexes of our DNA, our DNA, which are this double helix structure that you see that's very memorable in the scientific world. And so DNA, we can break it down even further, contains our genes. And so that's what Dr. Savage has been mentioning, which many of you may know. And those genes are instructions for life, they say. And each gene has a different name. We hear RTL1, TERT, TNF2, those are different genes. All right, and so I guess sometimes that may be a little difficult to remember. So to maybe put this in an analogy that's a little easier to remember. So if you think about our genetics kind of with this analogy in mind, if you think of our whole genome as a book, often I think of it as a recipe book. It's just that collection of all of our genetic information in one book. And then we can kind of correlate our chromosomes to what would be a chapter in a book. So our book would have, a normal book would have 46 chapters, or 46 chromosomes. And in those chapters we have different sentences, which we can compare to a gene. And so each sentence in that chapter is very important in making sure the whole chapter makes sense. Or if you think about a recipe, that the whole recipe makes sense and works properly. And so if we take this sentence here, the gray cat ran down the hall, that would be an example of a gene. And so actually, and I'll go into this in a little bit more detail, but each of those sentences and the letters in those sentences are important. And so here we see that a normal sentence would be, the gray cat ran down the hall. But if we change just one letter in that sentence, the sentence then reads, the gray cat ran down the ball. And that sentence doesn't quite make sense. And so our genes work very similarly in that if we have just one letter change, or another kind of change in the gene, which we'll kind of go over a little bit, that can change the whole meaning of the sentence or the meaning or purpose of that gene. And so then our bodies don't have a difficult time understanding that instruction or that gene, and that kind of leads to the changes in our telomere biology that we'll talk about in a little bit. And so I just kind of want to go over some of the types of genetic mutations that you can see. So what we saw just a second ago, the gray cat ran down the ball, was what we would be called a missense change, when there's just one letter change in that gene. Another type of genetic mutation we might see would be the gray-green cat ran down the hall, which would be an insertion. So here we see a series of letters. Sometimes it's just one, but it's a series of letters that can be inserted that then changes the meaning of the sentence. We can also have a deletion where one word is removed from that gene, or a portion of letters are removed from the gene. The gray blank ran down the hall. So what's running down the hall? I don't know, but it's gray. And so that's an example of a deletion. Next, we have a duplication. The gray cat ran down the hall. And these are pretty obvious here, but I hope these analogies are helping kind of some of these scientific words or medical terms that you hear or might have heard in genetics make a little bit more sense. And lastly, we have what we would call a nonsense mutation. And that's when we have a mutation in the gene that causes the gene to stop early. And so here we only see the gray. That's it. And so these are kind of the common types of mutations. There can also be larger deletions where you're missing much larger portions of a chromosome. So you could be missing multiple sentences in your chapter, or you can be missing even half of chapters, sometimes whole chapters, a whole chromosome. So there are a lot of different types of mutations, but these are the ones that I think are most important to talk about today. And so what happens when there's a genetic mutation? That's what we've been talking about. So normally, if we have a normal sentence, our gene is working properly, we have what we would call a normal gene, and all of our genes encode different proteins, and that's what Dr. Savage was talking about earlier. And so these proteins are important for maintaining our telomeres at the end of our chromosomes. And so if we have a normal gene, we get a normal protein. You can maybe guess what happens next. If we have a mutated gene, that often results in an abnormal protein, or sometimes no protein at all. With some conditions, there are overabundance of protein, which can also be a problem. But these, I think, are what are most relevant today. So abnormal proteins or no proteins, and that can cause issues with maintaining our telomere biology. And so here's the same image that Sharon shared with us earlier. I just changed the colors a little bit. But here you can see how all of these proteins work together to maintain our telomeres. And so the different colors here correlate to different inheritance patterns. So how many copies of a genetic change do you need to cause a telomere biology disorder? And so we'll go through those inheritance patterns a little bit later. But I just wanted to kind of put this all together. And so the next question is, how do we find these genetic changes? And we've already discussed whole exome sequencing, which is a type of genetic testing. But there are many different types of genetic testing that I kind of want to review with you all. So the first would be single gene sequencing. And that's when we look for errors in a single sentence in the book. And that's when it's often used, this is not used as often anymore unless there's a familial mutation, when we know that someone else already has a genetic change in a gene, and we go looking for that specific mutation or genetic change in a single gene. This often isn't used just because the cost of genetic testing has come way down in the last 5 to 10 years. And so many people say, well, if we're going to look at something, why don't we look at them all? And the cost doesn't often change. But single gene sequencing is an option. There's also the option for targeted gene panel sequencing. So that's when we look at a collection of sentences or a collection of genes known to be associated with certain clinical features or conditions. So in this case, you might have had a panel that looks at a whole bunch of telomere biology genes. So all of those genes that were in that complex that I showed earlier, you could have a panel looking at just those genes. Next is exome sequencing, which we discussed earlier. And so this is when we look for errors in the most important chapters or most important sentences in the book. And in a few slides, I'll go over kind of what that means. And then whole genome sequencing, which we also mentioned in the question we had earlier, when you look for an error in every single word and every single sentence of the book, all of your genetic information. And so I think the direction of genetics is really moving in the direction of whole exome sequencing, potentially whole genome sequencing as well, but right now, whole exome sequencing is the largest clinical genetic test we can offer, or the largest genetic test we can offer on a clinical basis. If you want to have a whole genome, often that's on a research basis. There are pushes for this to be in the clinical space, but you'd be more likely to be offered a whole exome opposed to a whole genome. And that's because, if you see here, the exome represents only 1 to 2% of all of our genetic information, which is wild to think about. Only 1 to 2% of the sentences in our book are responsible for encoding for those proteins that are responsible for our cellular functions in our life. And so the remaining 98 to 99% of this genetic information would comprise then the rest of the genome. And like Sharon mentioned, there's a lot of regulatory factors, there are potentially other genes that are in there to be discovered. There are many other functions to the rest of the genome, which we simply just are continuing to learn more about and don't know enough about right now to test for go through the whole genome and offer that on a clinical basis. But those are kind of the differences between the two. They're both very comprehensive tests, and there are some considerations for any genetic testing, which we'll review in a little bit. So I kind of want to go over some of the inheritance patterns. We've heard autosomal dominant, we've heard X-linked, so I want to go over kind of what those mean and put it into pictures. So this might be some of what you've already heard, what you already know, and so I'll be going through several different family trees, or pedigrees as we call them in genetics, to kind of explain the differences between each inheritance pattern. And for TBDs, this can get a little complex, because it's all of them, almost. So in autosomal dominant, as autosomal dominant inheritance patterns, we have 2 copies of every gene that we talked about earlier. And with autosomal dominant inheritance patterns, you only need 1 mutation in 1 copy of that gene, or 1 genetic misspelling in 1 copy, and the other copy is normal or not mutated. And so in these families, we see that multiple generations are affected, and that males and females are equally likely to be affected. We also see male-to-male transmission that can occur, and that's important a little bit later, but you'll see why I'm mentioning it now. And that each offspring of an affected parent has a 50-50 chance of being unaffected versus affected. So here we see a dad with one mutation on a chromosome, and the other chromosome is healthy and normal. And so he has a 50-50 chance of passing on the mutated copy or passing on the healthy copy. And so half of his offspring would be affected, and the other half would be unaffected. And these are the autosomal dominant genes for TBDs. In teal here, these are strictly autosomal dominant genes, but those in pink are both autosomal dominant and recessive, and we'll go over recessive next. I also want to mention what a de novo mutation is, and Sharon mentioned this a little bit earlier, but de novo simply means just new. So you're the first person in your family to have a mutation in this gene. So here we see 2 unaffected parents, both copies of their chromosomes are normal. And so sometimes we can have a genetic mutation that occurs in the egg or the sperm or shortly after fertilization that then is in the offspring of an individual. And so this is then most likely in every cell of their body. Sometimes it's in a fraction of them. But this is what we would call de novo, because they're the first person to have this mutation in their family. And the recurrence risk for these mutations is low across families. But what's important to know, and why I mention it now, is that once an individual has one of these mutations, that then follows that autosomal dominant inheritance pattern that we were talking about earlier. So although they're the first person in their family to have it, they can still they have a 50-50 chance of passing that on to their offspring. And for TBDs, that gene would be 10F2. That's a de novo gene. And before I move on, any questions? Does that make sense? I'm going to take the silence as a yes. All right. And so the next inheritance pattern that I want to discuss, which is a little more involved, would be autosomal recessive. And so that's when you have 2 mutations, 1 in each copy of the gene. in order to cause a telomere biology disorder. And so here, I'll go over some of the points about it first before reviewing the pedigree. But males and females for autosomal recessive conditions are equally as likely to be affected. The chance of offspring being affected varies based on the mutation status of both parents, because it requires 2 mutations. The greatest recurrence risk for autosomal recessive conditions is among siblings of affected individuals. Because you share, we're assuming you share the same parents, and so the recurrence risk is greatest there. And ethnicity and consanguinity, or marriage between families, may influence the likelihood of autosomal recessive disease. And so here, if we look at this pedigree, we'll see that both parents, they're both unaffected and what we would call carriers, meaning they have a mutation in one copy of their gene. And so each of them have a 50-50 chance of passing that on. And if you do some fancy math, you could see that about 1/4 of individuals are unaffected. So you have a 1/4 chance of receiving no mutated copy of the gene. You have a 50% chance of also being a carrier, so only carrying one mutation, and you would also be unaffected. And then there's a remaining 1/4 or 25% chance that you would be affected, that you would receive both the mutated copies from mom and dad. And the term carrier, some of you might be familiar with that from the prenatal space. You're often offered carrier screening in the prenatal phase. So we're looking for one copy of a mutation in one gene to see if you can pass it on to your children or offspring. And the telomere biology genes are included often on that carrier screening. So you may learn about a carrier status, but that doesn't always mean you're affected. And here are the autosomal recessive genes. The blue, again, are strictly autosomal recessive, but this pink, again, are both dominant and recessive. So some of these gene names you may be familiar with from your own experience. But I do want to return to this point that the chance of offspring being affected varies based on the mutation status of both parents. And so here I shared when both parents were a carrier, but I want to look at a different pedigree in which a father is just a carrier. And so in this case, if you have a mom who's not a carrier, or one parent who is a carrier and the other who's not, all of the children will be clinically unaffected. So you have 50% end up being carriers as well, and 50% are unaffected. But then if you have a parent who's affected with a recessive disease, all they can pass on is this mutated copy or this abnormal copy of a gene. So all of their children will be carriers, but if the other parent is not a carrier, then there's no genetic disease in their offspring. And lastly, I want to go over X-linked recessive inheritance, which, as the name suggests, are mutations carried on the X chromosomes. So females have 2 X chromosomes, XX, and males have an X and a Y chromosome. And so that's why we see the incidence of the condition of X-linked conditions are much higher in males than females, because they only have 1 X chromosome. And if they have a mutation in their only X chromosome, there's no other X chromosome to kind of pick up the slack, as we might say. So we see these conditions most often affecting males versus females. And so the condition can never be transmitted from a father to their son, because a father gives his Y chromosome to a son. And so you never see a father who's affected have a son who's also affected, at least as a result of the father. And so the chance to have affected offspring depends on 2 things: which parent is the carrier, kind of as I alluded to, or the sex of the offspring. And so here we see a mother who is a carrier, so she has 1 X chromosome that's normal and the other that's abnormal or mutated, and she's clinically unaffected, but has a 50-50 chance of passing on, as we've kind of seen in some of the other categories, this normal or abnormal copy. And so all of her daughters will be unaffected. There's a 50% chance one of her daughters will be a carrier, but for her sons, there's also that 50% chance that she'd pass it on, but one son will be unaffected, the one who does not inherit that mutation, and the other son who potentially receives this mutation will be affected. And so before I reveal which gene is the X-linked gene, pop quiz, which one is it? Dr. Savage mentioned it earlier. Does anyone know? Remember. You can be wrong. Just shout it out. It's the first one. All right, it's DKC1. That's the X-linked gene for dyskeratosis congenita. And lastly, I just want to show X-linked inheritance patterns when the father is affected. And so here, like we mentioned, an affected father can never have affected sons as they're passing on that Y chromosome. So not the X chromosome, they're passing on their Y, so they can never have affected sons. And both of their daughters will be carriers because they can only pass on that affected X chromosome to their daughters. But those daughters will be unaffected. So now that I've gone through all of that, we can kind of talk about the different types of genetic testing results. So these are some of the words you might have heard in clinic and might have read on your report: pathogenic, likely pathogenic, a VUS, benign. But maybe some of the other language that you're more familiar with would be positive. So pathogenic or likely pathogenic variants are what we call positive result. And that means when a disease-causing genetic change or changes were identified in a gene or multiple genes. A negative result would be this likely benign or benign category when no clinically significant genetic changes were detected. And lastly, a common category is this uncertain category. And in genetics, it's our least favorite category because it is so uncertain. And that's when a genetic change or changes were identified, but its clinical significance is unknown. And so I want to go into that a little bit more, especially as genetic testing is expanding into the whole exome space. You know, we all have genetic changes that make us unique, as Dr. Savage mentioned earlier. And so it's not uncommon to find genetic changes in our DNA. We all are walking around with different genetic changes. But whether we know what it means is another story. And so that's where this uncertain result comes from. And the more genes we look at with whole exome or whole genome, the more likely we are to find an uncertain result. So to give you an example of an uncertain result, or VUS, if we take this original analogy or this sentence, the gray cat ran down the hall, if we change gray to be spelled with an E, that's a genetic change, and the sentence still makes sense, but we're not sure if it impacts the function of the sentence overall in the cell. And so that's an example of a variant of uncertain significance or an uncertain result. And so in this case, testing the biological parents or sometimes other relatives for that specific uncertain result can help in determining its clinical significance. So if we find an uncertain result in an offspring who's affected and we test both parents and one parent has it and the other parent doesn't, and the parent who has the mutation is clinically unaffected and healthy, that's supporting evidence that this is just a normal familial genetic change that is probably not associated with disease. It doesn't rule it out, but it can be supporting evidence. And so that's kind of where testing parents can be helpful in determining, what an uncertain result means for your family. In the case of an uncertain result, future reanalysis may allow for reclassification to positive or negative as more individuals complete genetic testing and more research is completed. So I would encourage you, if you've had genetic testing in the past and did have an uncertain result, I think, we commonly in clinic have patients reach back out and say, I'd like to get a reanalysis. It's been about 5 years, and many genetic testing companies offer that as a service, generally free of charge. I don't know anyone who does charge for it. So I think that's a reasonable recommendation, especially if you're completing it on a research basis. Hopefully they're already doing that reanalysis periodically, but it's something that would be worth looking into because maybe over time we have more information and we can tell you if this result has been upgraded or downgraded to positive or negative. And I will say it's more likely to be downgraded to negative, but there is the possibility of being upgraded to positive. And more likely to be downgraded to negative just because we all have those changes that I was mentioning earlier. Lastly, and what's really important to know is that changes to medical management are not made based on an uncertain result alone. And that's something that is a common misconception in the medical community. And so we just don't want you to make any drastic life changes based on an uncertain result, on a result that could very well be downgraded to negative. And so in the case of an uncertain result, we suggest that you manage your care based on your symptoms. So to kind of go over some genetic testing considerations, some benefits, some limitations, and some other considerations for genetic testing. So some benefits include genetic testing may provide an explanation for the family health history. The results may assist with medical management decisions and may provide information for additional family members. Additionally, maybe in the context more so of DC and TBDs, there may be relief from uncertainty and anxiety regarding the presence of DC or TBDs in your family when participating in genetic testing. Additionally, information for individuals and families, it can be beneficial for decision-making processes. Some limitations to genetic testing would include a negative result is most informative when the familial mutation is known. And it took me, honestly, just a couple of reiterations of reading that sentence to really make sense of it. But if you know a mutation exists in your family, and if we look at that specific gene and we don't see any genetic changes in that gene, that's when that negative result is most informative, because we know there's a risk for that genetic change to be passed down. And if we don't see it, that's a very informative result and a positive negative result per se, right? There's no genetic change in that gene that we already know about running in your family. Additional limitations include that genetic testing at this time may not be able to detect all gene changes. And some companies vary based on what kind of genetic changes they look at. And some look at more or different types of changes than others. So everyone has a little bit different methodology there. And lastly, a limitation, which we just reviewed, is the possibility for a variant or a mutation of unknown significance or uncertain significance. An additional limitation that I wanted you to consider in the context of DC and telomere biology disorders is that the identification of a mutation in one of these genes indicates an increased risk, but it does not give you certainty of developing any physical or hematological findings associated with a TBD or cancer. And so that's one of the complex things about TBDs is that, and Sharon alluded to this earlier, it's just we have families that share mutations, but they can present completely differently in individuals and across generations. And so, unfortunately, it's not okay, we found this and this is what will happen. And so that is a limitation to genetic testing, particularly in the TBD space. Some additional considerations, and I'll kind of go over maybe some of the psychological or psychosocial considerations, but there could be a psychological impact to participating in genetic testing. In addition, a normal result could also give false reassurance. And I mean, maybe there is something there, but we just didn't find it, we just didn't look at it. So that normal or negative result might be misleading in some contexts. There could also be questions about insurance or employer discrimination. And I will say that there are laws in place that should protect you against discrimination with regards to health insurance, disability, and there's one more. I pulled the sheet later because I didn't put this on here. But there are laws that protect you against some discrimination, from your employer, but not against life insurance, long-term care disability. So I flipped that around. But we can talk about more of that in depth because I didn't review that here in this slide. But if you have questions maybe about how genetic testing relates to some of your protections with regards to insurance, I'd be happy to chat with you more about that. Some additional considerations include that, and Dr. Savage alluded to or mentioned this earlier, is that genetic testing for TBDs seeks to identify rare mutations for a rare condition. And so a large majority of individuals actually with TBDs, we don't know their gene. And we're learning more every day, and we know more people than we did 10 years ago. We know more about their genes. But sometimes, there might be a negative result, and that's why I suggest that reanalysis or an uncertain result. That's why I suggest, kind of investigating that reanalysis if you or other people in your family have an uncertain result or a negative result. Additionally, another consideration for genetic testing is discovering persons with an unsuspected affected or carrier status for DC and TBDs. And that's just because this presentation is so heterogeneous. And so you may find a genetic change, a mutation in someone in which is clinically unaffected, but they do have the mutation. It's hard to make sense of that. And I am nearing time, but I think it's important to review some of the familial considerations for genetic testing, and maybe we'll go over some family planning here at the end. But I will say, if you've participated in genetic counseling or testing, obtaining a family health history or building a family tree, or we call it pedigree, is a routine part of a genetic counseling appointment. And that's for many reasons, many of which might be a little intuitive that you can already guess. But this provides an opportunity to identify potential inheritance patterns, so that dominant, recessive, X-linked, and identify other at-risk family members who might also benefit from genetic testing. Additionally, this allows for genetic counseling referrals and or genetic testing recommendations to be made for at-risk family members. And this can help guide medical management, so screening and preventative care and support for at-risk family members. But I do want to mention here that the option to participate in genetic testing is up to the choice of an individual or their guardian. And so something that we hear a lot is, I know that I have this mutation in my family and I want my sister to get tested. There's a 50% chance she could have it, but she just won't do it. And as much as we love them and we care about them, the choice to participate in genetic testing is up to that sister. So as much as we love them and want to protect them, in genetics and specifically genetic counseling, we really want to respect the autonomy of every individual. And so we can't force anyone to get genetic testing. So that's something is a little complex, and I know that we all have the best of intentions of wanting our family members to also get genetic testing, but we unfortunately just can't make people get genetic testing. And I think there's a lot of considerations there, but I think one of the major considerations are the potential impact on family dynamics or the psychosocial impact on family dynamics when considering genetic testing. And people might have different thoughts going through their mind, but I did want to share some of those. And so some considerations might be disparities between affected and unaffected individuals regarding the amount of time spent with and attention received from family members serving as caregivers. And so that may be a worry for some individuals, preventing them from potentially participating in genetic testing. Additional considerations include feelings of being indebted to or obligation to family members regarding possible organ donation, intrafamilial, familial, or extrafamilial communication about DC and TBDs. And there are specific examples here that I won't read off for affected and unaffected family members, but these might be considerations going through other people's minds with regards to their reasoning as to why they want to participate in genetic testing or not. And these may be reasons that aren't shared with you, and is another reason to respect, your sister or your brother's decision to not participate in genetic testing. And so I do have some other slides on family planning and reproductive options for DC and TBDs. I do want to mention there's no one-size-fits-all option, and I want to send home that if these decisions are made around a table with your loved ones, you will never make the wrong decision for your family. And that's something I heard in clinic from one of my supervisors, and I really want you to take that home. For the sake of time, I won't go through the family planning and reproductive options in their entirety, but if you want to have a more informal conversation about that, if that's kind of the space that you're in right now or think you're heading towards, please reach out to me and we can talk more about these family planning options. But do know there's the option to potentially complete genetic testing for pregnancy, during that pregnancy, after that pregnancy, or even before the pregnancy, using what we would call pre-genetic testing or pre-implantation, genetic testing or diagnosis. So knowing the genetic status of an embryo before it's implanted. So there are options, and I'd be happy to talk to you about them. There are different limitations and risks associated with each. But just remember, there's no one-size-fits-all option. I'm not here to tell you one is better than the other. But if these decisions are made around a table with your loved ones, you will never make the wrong decision for your family. And so just reach out if you have questions about that. And with that, I do want to share a QR code to the DC TBD Diagnosis and Management Guidelines. Shameless plug, chapters 4 and 5 are on genetics and genetic counseling. And then I also want to share another shameless plug. The QR code on the right is for our DC and TBD Needs Assessment Study at the NCI. And this is a study that you'll hear a little bit more about next in a few presentations. But we're doing this study so we can get better at providing clinical care to you all and your families and your loved ones. And so there's a QR code there that will link you to a survey, and you also have the option to participate in an interview. So I think I'm a little short on time, but if you have any questions, you can ask or catch me later. I'll be here for you tomorrow.